Okay, let's begin. I'm Patrick Sullivan, and I'm here with my co-authors on the paper listed here, Kerstin Lindblad-Toh of the Broad and Uppsala University, and Stephen Gazal of the University of Southern California. We're really pleased to talk to you about the highlights from this paper that's in Press and Science, projected to come into print toward the end of the year. The format is Kerstin and Stephen will both give a 20-minute talk, followed by 5 minutes of questions. Kerstin's titles and Stephen's titles are listed there, and I will follow up at the end with 5-minute presentation, 5 minutes of questions about some practical stuff about how to use this. With no further ado, we'll talk about how to ask questions in a sec, but I want to turn this over to Kerstin to give the first talk. Kerstin.
Sorry, I'm trying to get rid of the extra boxes on my screen. Can you now see my slides?
Yes.
Yes. All right, so I would really like to thank the organizers for giving me the opportunity to speak here. And I know that Stephen and Pat is talking about my very favorite things, how you can apply mammalian constraint to understand human disease. But today I will talk about the alternate things that can be done with evolutionary constraint, which means looking at genome function and genome evolution. So as you all know, the human genome consists of roughly 3 gigabases of sequence, and only 1 to 1.5% is coding. It's been known for quite some time that a larger chunk of the genome has been functional, but it's not been easy to mark up all the functional elements in the genome. We also know that 90% of disease-associated regions are outside coding regions, which means that we really need to understand these regions better to understand human disease. So in 2011, we had a project called the 29 Mammals Project, where we sequenced 29 mammals and then looked across them for evolutionary constraint. And one of the key powers of evolutionary constraint is the branch length, which means that in the case of the 29 Mammals Project, we had a branch length of roughly 5 substitutions per site. So that means that when you look across all of these genomes, you will have a position mutated randomly about 5 times across the mammalian tree. And with this power, you cannot identify single individuals that are constrained, bases that are constrained, but we could instead find 12 base pair elements. As you know, we are talking about Zoonomia today, and that's also being called the 200 Mammals Project. For this project, we have about 17 substitutions per site, so we have much better power, and we can expect to find each individual position as constrained or non-constrained. Just to give this a comparison to what you do in human genetics, the NOMAD version 3 data set with 70,000 individuals has a branch length of roughly 0.17 substitutions per site, which means that you cannot detect constraint at the base level. You have to look at genes or possibly exons to determine what is constraint and likely functional. For this project, we sequenced 131 mammalian species and combined them with 110 existing assemblies. And this allowed us to get to a position where we have the power to look at single base pair resolution. With this data set, we generated a reference-free alignment with the aligner called Cactus, and this was driven by Benedict Patton's lab. Cactus is really good because it's a reference-free alignment. That means that you can take the 2 nearby species, such as human and chimp, and align them and deduce their common ancestor genome. And then you can continue to add additional genomes until you reconstruct the whole data set. So why do we care if it's a reference-free alignment? Well, it's because we want to be able to not just study the sequence that is in the human genome. Here on the left in the pink box, we have an element that has been lost in human, but is important in other species, and we might want to compare that. we might also want to capture everything for example, the dog, and then that we will have more sequence in the dog than if we were just aligning it to human. In 2020, we published this data set. So we published a new genomes and the alignment of the 240 species and also constraint scores. Now we have been busy trying to analyze this data set in many different ways, and I'm going to talk about some of the highlights of this. So Bill Murphy's group took all the 240 species and resolved the tree, and they did this based on 500,000 near neutral sites across the genome, and that meant that we now know with high confidence how all these 240 mammals are related to each other. They also looked to see where the diversification of mammals happened, and the result is that it happened both before and after the KPG boundary, where the big mass extinction happened. And so it's been a progressive diversification. For about 20 of the genomes, we generated some extra data to be able to look at chromosome level analysis. And this is work by Harris Lewin's group. And what you can see is that there are roughly 20 chromosomes in the ancestral mammals that lived for 100 million years ago. And you can see that a number of chunks are bigger than you would expect by random chance. So there's synteny across all the species for some large chunks, and one could speculate that those are regions of the genome that might actually be co-regulated and somehow needs to stay together through evolution. So we generated phylop scores, which is a measure of constraint. When we look at the data set, we see that more than 91% of the human genome is aligned to at least 5 species, but 11% is aligned across more than 95% of species, and this suggests that possibly all these regions might be constrained because they are present in all the species. However, when we look at the likelihood of something being significantly constrained at the 5% FDR level, we can pinpoint 3.5% as constraint at the single base pair level. If we are willing to allow some false positives, but getting a bigger data set, we can look at 20% FDR, and then close to 8% of the human genome is under constraint. Looking at the curves of constraint scores from neutral regions and the rest of the genome, we can predict that at least 10.7% of the genome is under constraint, even if we cannot yet detect it fully. And that, of course, matches very well with the 11% that is aligned. We also see that constraint base is clustered together. 80% reside within 5 base pair of another constraint base, and 30% are in 5 base pair or larger elements. If you look at loss of variant functions, you will see that the phylop score is often very high, overlapping that position, and then it falls off as you move away in both directions. Pat looked at 100 KB windows and could find 53 bins with significantly elevated constraint, more than 17% versus 3.5% genome-wide. And what we see are large regions containing a lot of regulatory sequence. And this includes the Hox clusters, which are known developmental transcription factors. If we look at coding regions, we can see that there is plenty of constraint in position 1 and position 2 within codons, but in the 4-fold position, there is much less constraint. If we look at the methionines, we see that methionines, that is the start of a protein, is more constrained than other methionines in the protein. And if you look at cysteines, those that make disulfide bridges are more constrained. So all of this makes a lot of sense. We can see that you have increased power to now detect non-coding regions as well. If you look at the fold enrichment of constraint bases, you obviously see that this is highest in coding regions, but it's also present for UTRs and different types of enhancer elements and such. Early on, when there was mouse and rat and human and dog to look at, people identified ultraconserved elements, and these were typically 200 base pair or larger, and in principle identical. Here we expanded this analysis to look at positions or look at elements that are more than 20 base pairs and are identical in more than 98% of the species. This means that we now find some 4,500 zoo UCEs, which are likely to be highly conserved enhancers, many of them. We also see a lower minor allele frequency in top med for these elements. We were also noticing that there were a lot of constraint elements that were lacking any kind of other annotation. So we termed these unicorns and annotated into genic constraint regions. There's more than 400,000 of these. They are between 11 base pairs and 1,300 base pairs. They are deficient in single nucleotide variants, and they have a lower minor allele frequency than random regions. And I'm showing you an example here. This unicorn is about 380 base pairs and is sitting in a gene desert on chromosome 10. There are multiple link RNAs in the region, but the closest protein coding gene is the GATA3 gene, which is about a megabase away. GATA3 is interesting because it's an immune regulator. And intriguingly, within this particular unicorn, we have a SNP that's highly associated with asthma, eczema, and eosinophil activity. So we can have a relatively high likelihood that this variant is functioning through this unicorn. We also wanted to see how the constraint, which is basically independent of cell type and time point, would compare to function as detected in a cell line of some type. And the Kircher et al. Paper had studied 20 promoter regions, and in this case we're looking at the LDR receptor promoter, and you can see that the phylop scores on top and the functional variant effect in red are strongly correlated. If you then zoom into this region and you apply ClinVar data, what you see is that the 4 orange SNPs that have been annotated as being pathogenic in ClinVar also have very high constraint and functional annotation, while the variant further to the left has uncertain effect and has very low constraint. Greg Andrews' group also looked at a large number of transcription factors. And what we can see that in the panel B, that CTCF is very constrained in some sites in red and not very constrained in other sites. And this appears to be the same with the many other transcription factors we looked at, where there's similar patterns that the constraints correlate, the constraint position correlate much better with the binding motif. And if we look over on the right, you will see that CTCF sites that are constrained are really shared across mouse, rat, human, macaque, and human, while the non-constrained position is not very constrained, although there are some similarities between macaque and human. We also know that the genome contains a fair amount of transposable elements, which was previously called junk, but it is of course not junk. It is in fact a source of novel innovation. So when we look across the tree, we can see that the genome size is correlated with TE content, not very surprising. But we also see that different families are present in different species or clades of species. And intriguingly enough, when we look at our constraint positions, we see that 11% of these are in transposable elements. Sorry. And when we see where the TAs that drive innovation are are primarily in primate binding sites. You can really see here at the bottom that there is evolution based on Ts in primates and therefore hence in the human genome. We next wanted to try to connect genotype to phenotype, and you can do that in different ways. The olfactory receptors are known to have large numbers of duplicated genes. And if you look at what species has how many genes, you see that the elephant has more than 4,000 genes, olfactory receptor genes, while killer whales, for example, have very low numbers. We also noted that there was a correlation with terminals, and terminals are features in our nose that allows us to smell. So it makes a lot of sense. Next, we looked at hibernation and the coding sequence of some 16,000 genes. Looking across the tree, we could identify 22 hibernators and 150 species that were definitely not hibernators. And we could see that some 11, at 5% FDR, some 11 genes were evolving slower and 11 genes were evolving faster in hibernators. And these are genes involved in temperature regulation as well as mitochondrial repair. So those are actions that are likely to be important in hibernation. Irene Kaplov developed a method to actually look at the non-coding parts of the region. So what she did was that she took the tree of mammals, and then she had open chromatin as taxic data for 5 species, including human. And then she performed a machine learning exercise to be able to predict open chromatin across the tree, and then she could correlate this with particular phenotypes. And in this case, we looked at brain size, where one of the top genes that come out is macro-D2, which has been implicated in micro and macrocephaly and intellectual disability. If you look at vocal learning, the TSH-Z3 and other genes are enriched in speech disorders. So I just want to summarize by saying that genome function can be studied very in-depth by comparative genomics, and that we see that roughly 10% of the genome is functional, and that roughly 50% is currently unannotated by efforts such as ENCODE or GTEx. In terms of genome evolution, we can identify highly constrained regions, but we can also identify accelerated regions. And I did not speak to this, but as part of the paper, there's also a description of accelerated regions, either in humans or in chimpanzee. And obviously, these are regions that could potentially be those that make us human. We can also do genotype-phenotype associations for both coding and non-coding regions. And it can, of course, be applied to human disease, and Stephen and Pat will talk about this. And before they get the stage, I just want to say that even for cancer, the non-coding, the constraint scores are highly informative. My group has looked at tumor mutations that fall in non-coding constraint mutations, and for multiple cancers, including medulloblastoma and glioblastoma, we can identify genes that tend to be differentially regulated and hence form driver genes using non-coding constraint mutations. So first, I want to thank all the people who contributed samples, and it's a huge number of people. And in particular, shout out to Oliver Ryder at the San Diego Zoo, who was a major contributor. I also want to acknowledge the roughly 150 people in the consortium. In particular, Matthew Christmas, Irene Kaplov, and Diane Genereux, who's been spearheading this paper that I talked about now primarily, and then Pat Sullivan and Stephen Gazal and Jennifer Meadows, who have been spearheading the flagship 2 paper that you will hear about now. And of course, a big shout out to Eleanor Carlson, my co-leader of the project. And that's it. Thank you.
Kerstin, thank you very much. For questions and answers, you can either type something in the webinar chat or raise your hand to be called upon. Elkis Price has a Q&A. Can you see it, Kerstin, or shall I read it off?
It's not coming up. Yes, sorry. It was.
So Elkis asks, why does little brown bat have much larger percent constrained than other species?
So I think it looks a bit different for different species, and it's not, we can't quite know if it is a composition of genome quality and genome size, but in the case of the little brown bat, the genome is much smaller than the human genome, which means presumably that the fraction gets to be larger for about the same amount of constraint bases.
Okay, thank you. Other questions for Kerstin? I'm trying to see. Other questions? Well, let me try one. So you, of course, you were the driver of the 29 Mammals project in 2011, and now Zoonomia. What's next?
A large number of primates. We see, as Stephen will allude to in much more detail, we see that the enrichment is higher in primates than in humans. You won't ever, there's not enough diversity in the primate lineage to get to single base resolution in primates, but we should be able to do at least twice as well as what we are doing now in terms of detecting smaller elements. And that project will also, I think, start to allow us to see element turnover and element creation in different parts of the primate lineage. So we're looking forward to working with a large number of primates, around some 350 of them.
Wow, outstanding. And there's a question in the Q&A box from Mark Adams. Did the results show?
Okay. Can I just add? I think the other thing that we would love to see is for all of you out there to really use the constraint scores in human disease. So go ahead and use it. So.
Thank you.
All right, sorry.
And Mark Adams asks, did the results show evidence of convergent evolution?
I think you could argue that the studies for hibernation might be showing convergent evolution, because it seems like the switch has happened multiple times between hibernators and non-hibernators, and sometimes the genes will be different and the pathway the same, and sometimes it will be convergence on different genes.
Thank you. Jonathan Coleman asks, I may have read this wrong on the slide, but chromosome X seemed largely consistent from the 100-million-year-old mammal to humans. Is this correct? Does this indicate that the whole genetic component of chromosome X works together?
So yes, it's correct that it is inherited together, but I don't think it is. I think it's a function of you need to have the whole X chromosome as it is the one where because it has the function that it has. I don't, as a sex chromosome, not because all of the genes there have to hang together for other biological reasons, is my guess.
Okay. And then the last question we'll take is from an anonymous person. Can you point to how we can access these valuable data?
I think Pat will talk about that in some more detail, but the key is to have it, among other places, on the UCSC genome browser.
Right. So there's two versions. I will talk about the gene-related stuff, and then I will post later the files that I'm using that actually is the base pair resolution, which will have a score per base. Okay, Kerstin, thank you very much. Great talk. And next, it's been really fun for me to get to know Kerstin over the last bunch of years working on this, and Stephen as well. He's been just a fantastic collaborator and amazingly into rolling up his sleeves and getting shit done. So it's been fantastic to work with him.
Thanks,
so Stephen, why don't you go ahead and kick it off?
Yeah. Okay, so hi everyone. So I'm Steven Gazal. I'm an new assistant professor at University of Southern California. So it's really fun to present here. I've presented here like a few times when I was a postdoc with Alkes. It was also like really fun to work with this consortium, like during, especially during COVID. It was kind of like my COVID project. So the one I was working like alone by myself in my bedroom. So I feel like really emotionally attached to its project. Anyway, so basically, like Kerstin, I've shown you like how great is the resolution of this new constraint score to understand genome function and evolution, and I'm going to talk about how to use this score to improve our understanding of human disease. So I don't know, a few intro slides. I know like some of you are like really familiar with that, but basically I will talk mostly about GWAS. And so as you know, GWAS have changed our understanding of disease genetic architecture. Like it shows that human complex traits and disease are extremely polygenic, which means that there's like hundreds to thousands of common regulatory variants with weak effects that confer a weak increase in disease risk. So GWAS has been like pretty successful to find associations. On the left, it's like usually what we observe when we look at the GWAS catalog. Right now, it's around about 400,000 associations that have been reported. But on the other side, when you look at the causal mechanism that has been highlighted, it's like really few. Like here's an example of a review that really show like a low number of associations for which we found the causal variant, and when we perform like functional assays to understand like the mechanism of the variant. And so why is it so complex? It's because when you zoom into an association regions, you see a lot of variants that correlates with each other because of LD. And so it's really hard just to pick the causal variant and to pick to understand its mechanism and find the target gene and so on. So in these last couple of years, like there has been developed what is called like post-GWAS studies, where we have like generated like a lot of functional data to prioritize all these variants. For example, being able to know which one are more likely to be in a promoter, in an enhancer, in the coding regions, and so on. And so here I'm just like highlighting like few of the projects such as Nomad and TopMed, where we generated a lot of whole genome sequence data to be able to either understand how deleterious is a variant based on allele frequency, but also to know like which regions of the genome are like really constrained, like where we lack mutations, but has been like bigger functional projects such as ENCODE, GTEx, now our GVF to understand the function of regulatory variants. I won't go really deep into like this project, but what I wanted to highlight, and I highlighted in bold, is like the sample size of this project. It's like really like we're talking in terms of like thousands or 10,000s of data that are needed to build like a catalog of like what is important in the human genome. And so now I'm going to constraint. Like, so as Kerstin said, like Kerstin is a powerful predictor of function, which require way smaller data sets. So, here's like a slide from like the first LD-score regression paper in 2015 that shows that when we investigate a lot of functional annotation, the one that is the most enriched in disease heritability is an annotation construct only using 29 mammals. So the enrichment was around like reported as around 13 in this paper. Since then, we made some like, I don't know, methodological improvement showing that it was a little bit upward biased, like the true value is around 10. But anyway, I think that's technical detail. But I think the punchline here is like it's really informative, and you get that information by using only 29 mammals. And so there are other advantages of using like this constraint annotation among others. So first, I'd say it's required small data sets. But the second one is that you have a lot of common variants that are constrained in mammals. So here, like using this annotation, it's 2.6%. And so we also have like this view of like everything that is constrained in the human genome. It's impossible to have a variant that reach a common frequency in the population. But it's actually not the case. You have a lot of common variants that are constrained in mammals. Another advantage is that constraint can be base pair specific, especially at the Zoonomia consumption. It's not subject to LD, such as the QTL, where we have like a lot of a lot of variants that are tightly with each other. Here you can reach like this base pair resolution, and you don't have like the issue of, for example, finding the good boundary for an enhancer and things like that. And the last thing, which is nice, is that constraint is agnostic to cell type. So it's given information which is non-cell type specific, which could be like really hard to handle like when you look at functional data. And so despite all these advantages, like since this paper came out, like really few paper leveraged constraint in post-GWAS studies. So here I'm like highlighting like the most cited post-GWAS reviews and like 2 recent reviews in high impact paper, and none of them like mention constraint as a tool to like to leverage to analyze GWAS data. And so like our goal was just to and of this project and my analysis in particular was just to validate the use of Zoonomia constraint scores in post-GWAS studies. And so what we did is we considered like these Zoonomia scores that Kerstin introduced. And so basically we used 2 types of score. We used a phylogeny score in mammals. So these are the scores with base pair resolution that were computed using like the 240 mammals. And we also like paid attention to what was constrained in primates. So in Zoonomia, we had like 433 primate species. There's not enough branch lengths to run phylogeny. So we run FastCon, which is a window-based approach, and that will give us constraint in primates as a small window resolution. And so we used, we analyzed these annotations with LD-score regression, which is a tool I know like most of you are really familiar. And so what the goal of stratified LD-score regression is that it's taking the GWAS summary statistic of a trait, for example, schizophrenia. It's taking like all the SNPs and not only like the top GWAS hits. It takes a reference panel for which we have sequence data, so that we can model the LD with the genotype SNPs and also the missing SNP, and a bunch of functional annotations that will be analyzed together at the same time. And so as output, you have like enrichment of heritability as it's represented here. And so in this talk, I'm going to focus mostly on like 2 metrics. The main one is the heritability enrichment of this binary constrained annotation. And so basically the enrichment is like the fraction of heritability that the SNPs in the annotation explain, divided by the fraction of common SNPs that are in the annotation. So for example, let's say that a constrained annotation contains 1% of the common SNPs in Europeans. And if this annotation explains 10% of the variance of the trait, then it will have like an enrichment of 10. And another metric, which I will use only in one slide, is something called like the joint effect of an annotation. And so basically it means that in this framework, because we're analyzing a lot of annotations together, and because a lot of these annotations can correlate with each other, we are able to say like to compute its conditional signal, which means like which it is effect conditional to all the other annotation in the model. And it will be like, in our case, like really useful to find which annotation is leading the signal among all these annotations that correlate with each other. And so we're going to apply this tool in 63 independent disease and traits for which we have a large sample size. Okay, so the first thing we did is just to validate the constraint score, and just to validate that the higher the constraint score is, the more enriched in heritability you are. So what we did is we divide the genome in 100 decile, and in this each 100 decile we take the common SNPs that were falling in these annotations in these scores, and we look at heritability enrichment. And so what we observe pretty nicely is that the more constrained you are, the more enriched in disease heritability you are. So this is the what's labeled in the y-axis. Then you're decreasing to have like no enrichment. And so what was really interesting to observe is that for mammal, it was like the 4 beans that were like significantly enriched, which was consistent with the threshold that Kerstin mentioned, that 3.54% was of the genome was constrained in mammal with FDR 5%. And for primate, it was a little bit more. It was more about 5%. And so what we did for the next of the analysis is we considered these 2 annotations, and we just took the SNPs that fall in the top 3.54% of the scores. So the next thing we did is just to validate the resolution of our score. And so basically, we plotted the heritability enrichment as a function of the distance to a constraint base. And so what we observed for mammals is that we were able to observe this base pair resolution, where we have like higher enrichment at the constraint base, but lower enrichment when you get further away. And we have like even like a significant difference in enrichment between like the -1 and +1 base pair compared to the current base pair. For mammals and for primates, we're able to validate the fact that our score are like computed like around like windows that are like in the 10 base pair range. So that was like really reassuring to see like the consistency between what was defined before and on human disease. Okay, so here are like the results of the Zoonomia heritability enrichment compared to other annotations. So we compared to other constraint annotation in lighter blue, the Zoonomia annotation in dark blue, and to what we consider as like the most relevant functional annotation of the human genome in gray. So we have non-synonymous, anchor tree, promoter, and enhancer, coding regions, FineMath, GTEC, EQTL. And so what was amazing to observe is that 2 Zoonomia annotations are like the most enriched annotation. It's even more enriched, for example, than non-synonymous, EQTL of GTEX, and any anchor tree annotations. We observed that our annotation, for example, in primate was more enriched than the previous one, computed like with less individual, and the same using our constraint annotation in mammals. But as I said earlier, because all these annotations correlate to each other, right? A lot of bases are like constrained in like multiple alignments. A lot of constraint bases also like fall in the coding regions. What we did is we used like this conditional effect to see which annotation was driving the signal. And so I'm reporting here the p-value. And what we observed is like a huge significance for the constraint annotation in primates. The p-value is around 10 to the minus 50. It's way much more enriched than any other functional annotation, especially FineMath, GTEC, EQTL. And again, I think it's kind of like amazing just to have like this kind of power and new information based only on like 43 primate sequence compared to all like these other annotations that I don't know required way much more data. Okay, now next we compare these enrichment in different type of traits. So first we replicated the results that enrichment of constraint bases in immune traits is lower. So enrichment of constraint bases are lower in immune traits. We investigated what was going on for a brain disease. So I've listed here like what I consider as brain disease, and we didn't find any kind of like significance. And the reason for that might be probably because constraint bases involved in brain do not reach high frequency and do not become common in the population. And this was kind of like validated here. So I'm doing like some kind of like sneak peek of like what's going on with like Pat's talks. And so basically what we did is we took like all the genes. I mean, what Pat did is he took all the genes and he looked at the fraction of their like coding regions that were constrained. And then for all these genes, we looked at the enrichment of the SNPs that were linked to these genes. And so what we observed is that genes that are like highly constrained are highly enriched in heritability of brain disease. And so this is consistent about like, I don't know, like other works that show that genes, for example, with high PLI score or like with more like with high constraint in humans were also more enriched in brain disease. Okay, so now we dig into like these really interesting results, which was that enrichment in primates, which didn't have base pair resolution and which considered like 5 times less species, provide higher functional enrichment. And so what we did is we compare like different enrichment of bases that were constrained in mammals, in primates, in both, or only in mammals, or only in primates. And so basically what we observe is that bases that are like constrained only in mammals are like really not that enriched in disease heritability compared to bases that are only constrained in primates. And then what we did is we stratified these variants based on their function. So interestingly, when we look at exon, we observed that we have like slightly higher but not significant enrichment for bases constrained in mammals, and this can be explained either by the fact that the genetic code in exon region has base pair resolution, or maybe just because like the function of the gene are kind of like of the protein are kind of constrained across species. But what was super interesting to observe is that like these enrichment like were like really higher in regulatory regions, for example, regions that were constrained only in primates and in promoter were like way more enriched than regions that were only constrained in mammals, in promoters. And we have like the same pattern for enhancer, which kind of like highlighting that constraint in primates might be more relevant to analyze regulatory regions than constraint in mammals, probably because regulatory regions, like regulatory regions are mostly shared across primates, but not across mammals. Here is the last plot to highlight what are the enrichment for constrained regions which do not fall in these free annotation, and also like in FindMap GTech EQT. And what was interesting to observe is that we still have some significant enrichment for this region, which kind of like shows that constrained information could be also valuable for regions without any other functional annotation. Okay, here I'm moving from common variant to common variant and low frequency variant to look at the expected squared effect sizes of constrained variants as a function of their allele frequency. And so let me start with like the blue line, which are like the constraint SNPs. What we observe is that these constrained variants have similar effect sizes from like 5% to 50%, and just due to flattening effect, due to negative selection. But what we can observe after is that we have like an increase of effect for lower frequency variant. And it's kind of like suggests that, I don't know, maybe we can use constraint variants to constraint information to prioritize rare variants in whole genome sequencing studies, especially in non-coding regions. So when we split this blue line into like coding SNPs and non-coding SNPs, we observe like a huge effect of effect sizes for coding SNPs, which is consistent with what we know about rare variants with high effect sizes. They tend to be non-synonymous or loss of function variants. But what we observed for non-coding variants, it's a less impressive compared to the red one, but still like higher effect for rare non-coding constraint variants, which also can, I don't know, highlight the fact that we can leverage these constraint information for rare variants, but not only for the coding one, but also like for the non-coding one. Okay, I start running a little bit out of time, so I just want to say that we finally, as an application, leverage these new constraint score to in functionally informed fine mapping studies. So we use these polyfun method, and the idea was just to compare like fine mapping results in analysis where you give prior based on functional annotation, and like what are the results when you add like these Zoonomia scores. So really briefly, I will highlight the results in the FT locus. I'm highlighting these results first because it's like one of the most famous example in GWAS, and also because we know pretty well like the causal SNP, which is RS 1421-085. And so what we observed is that basically when we just add the Zoonomia scores, we were able to increase the probability to be causal from 0.13 with no functional annotation, 0.58 with like default annotation, and 84 with the Zoonomia score. And it's because this variant is extremely constrained in both mammals and primates. And what was also interesting to observe is that the gene FEO, which is not the causal gene, is not really constrained when we look at the fraction of bases of its exon and promoters that are constrained, but the two causal genes, IRX3 and IRX5, have a lot of were more constrained. So it's kind of like suggesting that enhancer, like bases that fall into, for example, enhancer and that are constrained, are more likely to target a gene that is itself very constrained than a gene that is not constrained. Here it's a last example where just with the Zoonomia score, we're able to flip the probability to be causal between two SNPs, like with the default approach. Fine mapping highlighted the sentinel SNP, so the SNP with the highest p-value, the lowest p-value. But by adding the Zoonomia score, we highlighted like the other one, the one that has next, but with a smaller p-value, higher, sorry. Okay, so I'm going to stop here. So the conclusion is that Zoonomia constraint annotation was the most informative annotation to study human disease. They are more enriched in primates than in mammals. That was surprising first, but it can be explained by the fact that it's because human diseases are driven by common regulatory variants, and it looks more relevant to use conservation in primates than mammals to look at non-coding genome. We found some pattern of strong negative selection, and so suggesting higher effect sizes for rare constraint variants. We observed a low enrichment for blood immune traits, but higher enrichment around genes that are constrained for brain disease. And we, as an illustration, we show that it can improve fine mapping. So I'd like to finish this talk by acknowledging members of my lab, member of Alfred Price lab, and all the members of the Zoonomia consortium, including like all these ones with whom I collaborated a lot. Okay, that's it.
Okay, Stephen, thank you very much. We have time for a couple of questions, please. And as before, type your question into the Q&A box or the chat box if you would. I guess the first question, that I'd ask, Stephen, is, say I wanted to use this as part of partitioned LD-score regression. Is that an easy thing to do, or is it difficult, or what advice would you have for people that would like to give this a try?
Yeah, so we created like LD-score regression annotations, like for these two annotations. And so I don't know, we need to better communicate how to get them, but it's pretty easy to use it, right? It's one command line where you put the GWAS file and a bunch of files. Yeah, so it's pretty easy and straightforward to run.
Would you consider having like a link off of your USC website that would actually explain how to do this and give people the source files?
Yeah, you're right. I think I can work on that. Okay.
Because you also have the HC19 versus HC38 issue too, and don't want to mess that up.
Obviously. Yeah, so I converted like all the things in HC19 to annotate my variants.
Okay, yeah. Cool. Yeah, because I think people would be super interested in this. And we have a Q&A score. Did you try to challenge your score with LUF or PLI score for the haplo efficiency versions?
Yeah, so I think it's more like PAT section, but I think I've replicated this graph with like using PAT, LUF and PLI score, and it was like pretty much the same. Like, so it's the same kind of information.
Any more questions for Stephen? Yes. I am a bit of a newbie to GWAS, always worked on rare variants before. but could you briefly explain how one could use Zoonomia annotations to improve fine mapping?
Yeah, so I think there could be two approaches. I think the first one is you did as I've done. So let's say you have the genotype phenotype data, you can run fine mapping yourself. Then you can use this tool, which is Polyfun, which is a tool for which you give a bunch of annotation. It estimates the priors to give to each variant based on this annotation using like an even and odd chromosome type of thing. And then it runs fine mapping by prioritizing each variant based on these functional priors. This could be and this is how we did that. The second option is that you can, let's say you have like vanilla fine mapping results with like all the variants in the credible set. You can like overlap the probability to be causal with the Zoonomia score, and then you can manually prioritize the ones that are like really highly constrained. Because if you find like, for example, two variants are in really high LD, they have like similar probability to be causal, but one is constrained, and you can have this information because, like I said, like constraint is like base pair specific and doesn't suffer like this LD kind of approach. Then maybe you can say, oh, it's more likely to be this one than this one.
Okay, Stephen, thank you very much. If you could stop sharing, I will take it up for the last part. Okay, very quickly. The supplement of our paper is the key thing to look at. It's 90 pages long. It's divided into essays about specific topics, and only the conclusions from that actually make it in the main manuscript. Jinziki was here at UNC, and I looked at constraint and copy number variation, and we found that it actually was a really good way to prioritize copy number variants. So for example, constrained bases had the difference between schizophrenia cases and controls was 10 to the minus 19 versus sort of 10 to the minus 3, or even at best 10 to the minus 14, using sort of a more conventional idea. So we believe this is useful there too. Next, there's a big section, which was really a lot of fun to do, working with Naomi, Yun Lee, Laura Huckins, Stephen, and others, basically to look at PRS. And we found that using constraint to prioritize the training set, the SNPs in the training set, improved accuracy, and that constrained SNPs actually had a lot more per SNP information to contribute to the PRS accuracy. So we believe it helps there as well. See see that part of the supplement for details. Next, we worked on creating a gene-based measure of constraint. There's a big section on its creation and then on its evaluation. We of course compared it to PLI and LIF as well. And in the end, I did 7 different things, including a complicated model that required 24 hours to run. And in the end, it was really just a fraction the simplest measure appeared to be the best, which is merely the fraction of constrained bases in this fraction of constrained CDS bases divided by the total. So if no CDS bases are constrained, then it's 0. If they all are, it's 1. It's also useful to look at this as a decile. And you can read the details, but we believe that it had some excellent properties in comparison to the other measures that are out there. I forgot to mention this, that we actually have a complementary, there's an essay in there that compares what evolutionary constraint gives you compared to human genome surveys. And there are pros and cons of each, but in essence, they're complementary. And then the one thing I will close with is we did a gene set analysis of genes in the top 5% of constraint, the most constrained genes in the genome. And then we also looked at the least constrained. And the gene set analysis I thought were fascinating. If you look at the least constrained genes, the Go terms that came up were for defense response, taste and smell, and skin development and keratinization. There was nothing synaptic about that at all. In the top 5% of genes, the most constrained in the genome, it was basic embryology, the creation of essentially every major organ system, cell cycle, basic stuff, and also a ton of synaptic annotations. Effectively, the top 5% of genes have to do with how you make a mammal. And bottom 5% are genes that function at the boundary of a mammal and its environment, and that have a lot to do with how that mammal fits into its environmental niche. And I think this actually has a lot of implications for our work. If you study a skin disease, constraint might not be something that's the most interesting to you. And that was at least partially consistent with the graph that Stephen showed before. Okay, and how to get the data? If you look at the chat box, I gave you 2 ways to get it. Oops, I went too far. If you look, this is my version of it, that this folder contains the individual chromosome level, the actual base pair positions. Read the README, take a look at the README, it'll explain everything. And these 2 are bed files that are merged to basically give you the regions of the genome that are under particularly high constraint. Alternatively, you can go to this track, the 241, the cactus alignment for Zoonomia that's available off UCSC. And I've also, in terms of some of the key stuff, if you search for, if you go to Figshare, Sullivan gene matrix, you can get this. This is, there are 2 files. One is the actual 1 row per gene. Rows are genes, columns are information about the gene. The README explains everything, gives citations. The fraction of the coding sequence that's conserved across all mammals is this variable. The fraction of the coding sequence that's conserved across primates is that variable. So have at it, learn stuff. Love to know what you find. Okay, so we've got a few more minutes for questions for anyone. Please, what can we help you with? While people are typing, I need to give my thanks. I want to thank Kerstin and Eleanor in particular for allowing me to spend the better part of 2 years in my Zoonomia sort of staycation sabbatical working on these data. It was a privilege, and it was a fantastic example of team science. All the crazy stuff that I learned was nuts, like exactly how do you get the temperature of a bat and how poorly phenotyped a lot of mammals actually are. It's pretty cool. It was pretty fun to work with you all. And we have an anonymous attendee thanking us. Any questions, anyone?
And again, a big thank you to Pat and Steve and all the other people who've been involved. The enjoyment has been mutual. So
it was an honor and a pleasure.
Same here, same here.
Mark Adams, is there an evolutionary model that would relate effect size, minor allele frequency, and constraint score? Cool question. There's a big section in the supplement where I described at least this. As you would expect, minor allele frequency has a complicated relationship with constraint score. There's a big section in the supplement on this. But in terms of an evolutionary model, Kerstin, Stephen, do you guys know?
Yeah, I think there's maybe like a paper from Peter Vischer and Jen Yang, like where we are able to express effect sizes as a function of allele frequency and like a selection coefficient. It's like in the form of like p multiplied by 1 minus p to like a power something which is linked to evolution. And so I proposed a model to do that by functional annotation, right? And so each variant in each functional annotation, we have like a different alpha parameter. So we can think about like using this model.
Yeah, I remember that now. That's pretty.
I think it's kind of like related to like the curve I show. Like they're like models for variants that are either constrained, encoding, either constrained and uncoding. Yeah.
Okay. We're at time. I'd like to thank my co-presenters for a couple of awesome talks, and thank you all out there for your interest. As usual, this will be on YouTube at some point in the near future. Thanks, everybody. We're done.
Thank you.
Bye, everyone.
Bye, everyone.